Hello and welcome to the NIE Sustainability Learning Lab video series on water quality. Today, Grace Wee and I will look at nutrient levels in aquatic ecosystems and also demonstrate how to measure certain forms of macronutrients, nitrogen and phosphorus.
Prof, what do you mean by macronutrients?
That's a good question, Grace. In aquatic ecosystems, macronutrients are nutrients such as nitrogen, phosphorus, and potassium that are available in relatively high quantities, but also are essential for proper plant and algae growth. For example, phosphorus is readily available in soils and sediment and is very, very important for plant growth. Now, the phosphorus in the sediment and soils that are important is actually in the form of phosphate, and the phosphate helps to promote photosynthesis, respiration. It's important in cell division and also energy storage and transfer. In the case of nitrogen, nitrogen is also quite readily available in soils and sediment. However, another important source of nitrogen actually comes from our atmosphere. About 80% of the air we breathe, in fact, is nitrogen, and so the air is an important pool for nitrogen. The plants and algae will use the nitrogen in photosynthesis, and also the nitrogen helps to promote healthy plant growth and keeps the nice dark green color on the leaves of plants. Very often in agricultural areas and also in urban areas where we have parks and lawns, people will spread chemical fertilizers with macronutrients in them, so nitrogen, phosphorus, and potassium, to help promote crop growth or lawn growth. Now, unfortunately, too much of a good thing can actually be a bad thing. So in an agricultural area, for example, if you put too much fertilizer on the field, some of that fertilizer will not be used by the plants, and so the nutrients will then leach out from the soil and go into local waterways. In that case then, the algae in the water will happily take up the nutrients and will start to grow, as we clearly see in this graph. On this graph, the y-axis shows chlorophyll A concentration in milligrams per cubic meter as an indicator of algae activity, and on the x-axis we have phosphorus concentration, also in milligrams per cubic meter. We see that as the phosphorus concentration increases, algae concentration also increases. The process of elevated nutrients in a water body due to human activities is known as cultural eutrophication. And this can be bad news for a water body. So what happens is, initially we start to see green algae growing because they have high nutrient levels, and eventually this forms a green scum on the surface of the water or an algae bloom. The green scum will reduce the amount of sunlight that can go into the water, and eventually then the green plants below will die because of a lack of sunlight. In addition, because algae also are living things, eventually the algae will die, and so the algae dying combined with the green plants dying will start to create a problem. That material starts to decompose, which reduces the oxygen levels in the water because of the decay and oxidation process, as we saw in that previous video on dissolved oxygen. So now we have a problem with low dissolved oxygen levels in the water due to cultural eutrophication. This can be a problem for fish and other aquatic organisms.
That's interesting, Prof. But what concentrations of phosphorus and nitrogen in the water will result in eutrophication problems?
That's a good question, Grace, and a fairly complicated one. It really will depend on a number of factors. It will include factors such as the size of the water body, whether the water is flowing or still, and whether the water is freshwater or saltwater or seawater. In general, however, in freshwater, scientists have suggested that a total phosphorus level greater than 0.042 milligrams per liter and a total nitrogen level greater than about 0.3 milligrams per liter will lead to eutrophication problems. Now that we have a basic understanding of both the benefits and the detrimental impacts of macronutrients, Grace, could you show us how to measure nitrogen and phosphorus, please?
Sure, Prof. Today we will be using two different kits from Chemmetrics, one for phosphate and one for nitrate. Let's start with the phosphate first. After you collect your water sample, fill the sample cup up to the 25 milliliter mark. Add 2 drops of activated solution. Cap your sample cup and shake it to mix the contents well. Place the phosphate ampule tip first into the sample cup and snap the tip. Make sure that the ampule stays in the water. The ampule will fill up, leaving a bubble for mixing. To mix the ampule, invert it several times, allowing the bubble to travel from end to end. Dry the ampule and wait 2 minutes for colour development. To obtain a test result, use the appropriate comparator. If concentrations are high, use the regular colour standards by placing the ampule in between the standards until you find the best colour match. In this case, we can see that the concentration is less than 1. For low concentrations, use the tube comparator by placing the ampule flat end first into the comparator. Rotate the comparator until you find the best colour match. In this case, we can see that the colours falls between 0.1 milligrams per litre. If the colour of the sample falls between the standards, it is okay to estimate the sample value. Fill the reaction tube up to the 15 milliliter mark with the sample to be tested. Then, empty the contents of 1 cadmium foil pack into the reaction tube. Take care when handling this pack. Cap the reaction tube and shake vigorously for exactly 3 minutes. After this, allow the sample to sit for another 2 minutes. Pour 10 milliliters of the treated sample into the sample cup. Be careful not to transfer any of the cadmium particles over into the sample cup. Place the nitrate ampule tip first into the sample cup and snap the tip by pressing it against the side of the cup. Do not remove the ampule until it is fully filled. There will be a small bubble to facilitate mixing. Mix the ampule by inverting it several times, allowing the bubble to travel from end to end. You can then dry the ampule and wait 10 minutes for colour development. Place the ampule between the colour standards, moving it along the comparator until the best colour match can be found. If the colour of the sample falls in between the standards, it is okay to estimate the sample value. In this case, you can see that the concentration would be about 0.1 milligrams per litre. To convert to milligrams per litre nitrate, multiply that result by 4.4. Make sure that you collect and dispose of all water used in the testing to the chemistry department of your school.